Good evening, everyone. My name is Mulisala Panya. I'm a third year student in biochemistry and molecular biology. Today, I would like to talk about measurement and modeling of conjugative and mobilizable plasmid transfer dynamics. One of reproduction, one of reproductive process in unicellular organisms that occur naturally. That is conjugation. Conjugation is a process for transferring a gene from one bacterium to another come to course together. Bacterium can transfer gene called donor. Bacteria can receive called recipient, and recipient can receive plasmid become transconjugant. Why should we be concerned about this? Currently, many antibiotic-resistant genes use conjugation for expression. Healthy people are less likely to be harmed, but if someone who are physically weak and seriously ill may have a serious infection that can lead to death. And now, death from drug-resistant infection set to skyrocket. That's why we need genomic engineering to do something to solve this problem. For example, we can use conjugate for transferring CRISPR-Cas antimicrobial to edit the gene in the human's body. The microflip in the human body make up a microbiome that have more than 10 to 10,000 trillion, especially in the gut. So, biotechnology is important to adapt bacteria population in current. By conjugation, antibiotic-resistant gene will spread across bacteria population. We want to know how plasmid strain in the population at the time pass. Conjugation efficiency must be measured and modeled. And then the way we know it is the first we can make measurement be concrete in the lab. Actually, a measure how many cells we have in this stage of conjugation, and the other one is using mathematical in the computer model to predict and expand what happens. Research article in Google Scholar on 16 January 2022. We found 2620 studies from 2000 and 2021. From this page, I show two earlier research that are relevant to our subject. The first paper, scientists collect paper about conjugation efficiency. They need to know how different parameters affect to the rate of conjugation. Then they study across different families of conjugation plasmids. Scientists measure the rate of conjugation at that moment, but they never tried to simulation the dynamic of conjugation and the ratio of conjugations that are unstable over time. Scientists studied only conjugative plasmid, and scientists not studied related to mobilizable and conjugative plasmid. And the second paper, scientists use conjugative and mobilizable plasmid for effective transferring plasmid. They didn't know how many plasmid distribute as the time passed, and scientists distribute of conjugative and mobilizable plasmid that called RNCP and RNCQ. RNCP is conjugative plasmid and RNCQ is mobilizable plasmid. This is a leak of knowledge that I will need to fill. Our Our two goals are measure transfer rate of conjugative and mobilizable plasmid, and another one to develop a computational model that explains and predicts dynamic distribution of conjugative and mobilizable plasmid. Our project used donor and recipient. Donor is a host cell in our experiment were E. coli, ECGT2. ECGT2 ECGT2 contain um, can express TFP and requiring depth DAP supplementation. And recipient is Th5 alpha. Th5 alpha is a bacteria that can accept plasmid insertion exceptionally well, and the whole plasmid we use, that is PPSNU25. PPSNU25 contains ORIV and ORIT, fluorescent marker GFP, and selection marker chloramphenicol. And the other one is PRL 443, contains conjugation machinery OV and ODT, and selection marker kanamycin. And here, I will show how we measure conjugation rate. But the first, we mix between donor and recipient, then support on the nitrocellulose membrane. We suspend with PBS 1 ml and plate on selective plate. And the last, we count colony CFU per ml and calculate conjugation efficiency. This page, I will show relationship between the density ratio times conjugate approximately. We have colony count and calculation efficiency. But if you change a variable, for example, the proportion donor to recipient is 3 proportion. The result of mixing donor and recipient, the ratio of uh, 1000 to 1, 1 to 1000, and 1 to 1 show that 1 to 1, 1000 to 1 conjugate was the highest because recipient can receive 100% of plasmid. We will get relationship between donor and conjugation efficiency in the figure on the right. And another method, we have measure of growth rate of each bacterium. So growth rate measurement, um, I would I mean, this is a transmission of plasmids between cell growth and effect in the number of cells we can count. If the cell grows very differently, the result may be distorted. During the transfer of plasmid cells are growing and is dividing definitely. That's why we have to look at growth. We need to ensure that when we good efficiency transfer plasmid, it's not just from bacteria that multiply well. So the first one, we prepare bacteria from inoculation, single colony, and overnight. Then we drop in the 96 well plate and run on micropage liter, and we will got growth curve. Okay, this page show the measurement results of growth rate each bacterium. From the graph, the H5 alpha grows better than ECGT2. That each species has different K and R values. The K values indicate the highest OD and use for growth, while R value indicate good growth. Everyone will see the repeat growth of the H5 alpha can be seen with repeat weight in 10 hours, possibly due to too low temperature, too much humidity, or insufficient oxygen requirements. From this page, we will show code model by simulation. In the Google Colab. Google Colab is a platform that allows for Python's programming on web browser. And then we import Terrarium as TE. Terrarium is Python package that we can use for simulation and is a model for predict for experiment to determine conjugation efficiency among bacteria. This picture, we have simulated plasmid transmission in the bacteria population. The ratio we use is donor to recipient 1 to 100 was used to predict the dynamic distribution of conjugative plasmid. And, and then you will see many equations in this page. For example, equation I will show. Here, this is an example of an equation representing conjugate. This is a Python function. Python function is a function set of instructions that perform a specific task, where we can pass parameter into function. And this equation means conjugation rate depends on donor number of donor and recipient. D is donor, R is recipient, T is time conjugant, and KCJ is conjugation rate. Okay. Okay. For this page, I will show result of simulation um, qualitatively predict conjugation dynamics. Is a model for predict experiment between donor and recipient. This picture we have simulate transmission of one plasmid in bacteria population, the ratio of 1 to 100 of donor to recipient. I used to predict the dynamic distribution of conjugative or INCP plasmid. For this page, conclusion, we found the best ratio for transferring plasmid is 100 to 1. Growth rate have effect on conjugation efficiency. Third, we found a way to measure and transfer rates of conjugative plasmid by determining the conjugation efficiency. Fourth, simulation can describe and predict dynamic distribution of conjugative plasmid can be used. And in the future work, we will able to adjust bacteria population in the ratio of 100 to 1 for investigating their density and mating time. Second, we will use dynamic simulation for prediction of conjugation efficiency in other parameters. And the last, we will measure growth rate in solid and liquid frequency periods. And thank you, my advisor, and thank you everyone for attention.
ค่ะก็นิสิตใช้เว